Exploration and analysis of molecularly annotated 3D models of breast cancer at single-cell resolution using virtual reality. Published by BioRxiv. Dario Bressan, Claire M. Mulvey, Fatime Qosaj, Robert Becker, Flaminia Grimaldi, Suvi Coffey, Sara Vogl, Laura Küett, Raul Catena, Ali Dariush, Carlos Gonzalez-Fernandez, Eduardo A. Gonzalez-Solares, Mohammad Al-Saad, Aybüke Küpcü Yoldaş, Tristan Whitmarsh, Ilaria Falciatori, Spencer S. Watson, CRUKIMAXT Grand Challenge Team, Johanna A. Joyce, Nicholas Walton, Bernd Bodenmiller, Owen Harris, Gregory J. Hannon. Abstract. A set of increasingly powerful approaches are enabling spatially resolved measurements of growing numbers of molecular features in biological samples. While important insights can be derived from the two-dimensional data that many of these technologies generate, it is clear that extending these approaches into the third and fourth dimensions will magnify their impact. Realizing biological insights from datasets where thousands to millions of cells are annotated with tens to hundreds of parameters in space will require the development of new computational and visualization strategies. Here, we describe Theia, a virtual reality-based platform, which enables exploration and analysis of either volumetric or segmented, molecularly annotated, three-dimensional datasets, with the option to extend the analysis to time series data. We also describe our pipeline for generating annotated 3D models of breast cancer and supply several datasets to enable users to explore the utility of Theia for understanding cancer biology in 3 dimensions. Introduction. The ability to interrogate tissues at the single-cell level and to gather genomic and proteomic information at scale for the individual constituents of living systems is having an enormous impact. Single-cell RNA and DNA sequencing is revealing new cell types, enabling comprehensive catalogs of human and mouse tissues, and revealing genetic and phenotypic heterogeneity in tumors. Yet, most normal and disease development happens in a spatial context. The three-dimensional architecture of a tissue has profound impacts on its function, whether it be through the establishment of morphogen gradients in development or through the effect of local tissue niches featuring distinct cell compositions or signaling states. However powerful, measurements made on disaggregated cells or nuclei generally lack this critical contextual information. In cancer, there is a growing appreciation that the initiation of disease, its progression toward invasion and metastasis, and its response to therapy are all profoundly influenced not only by the properties of the tumor cells themselves but also by the characteristics of the tumor microenvironment, TME. A description of the relationships between a tumor and its TME requires not only an enumerated catalog of its constituent cell types and clonal lineages but also an understanding of their interactions and arrangement in three-dimensional space. The desire to understand development, tissue architecture, and disease in context has prompted a drive toward the development of methods for making multiplexed measurements on intact tissues or tissue sections. Such methods measure portions of the transcriptome or proteome or alternatively can quantify metabolites. Some are destructive, while others leave tissue intact with the potential for layering additional measurements, and though a few methods are compatible with multimodal measurements, generally the number of markers measured outside of the primary modality is rather low. MERFISH, StarMap, SeqFish, spatial transcriptomics, and SlideSeq can interrogate the expression of 100s to greater than 10,000 transcripts in situ, with most of these approaches operating at cellular or subcellular resolution. Imaging mass cytometry, IMC, Codex, CYCIF, 4i, and others can measure the expression of dozens of proteins simultaneously or serially. All of these are now beginning to produce new biological insights into the organization of tissues in 2 dimensions. For example, recent studies using IMC have revealed that the tissue organization in breast cancer is both informed by the subtype of the tumor and correlates with patient outcomes. While 2-dimensional, molecularly annotated maps of tissues represent an important advance and clearly provide added value to disaggregated datasets, 3-dimensional datasets with layered information comprising as many types of measurements as possible would maximize our ability to extract new biological insights. A recent analysis of gene expression in situ during mouse embryogenesis, which includes a time dimension, illustrates the power of such approaches. Lohoff et al. NAB Biotechnology, in Press. In general, spatial omics methods suffer from two main limitations. They usually operate only on thin two-dimensional sections of tissue, and their speed of acquisition is relatively slow. The first issue can be resolved by analyzing serial sections cut sequentially from an embedded tissue fragment, and aligning the resulting datasets in order to produce a coherent three-dimensional object. However, the slow acquisition speed means that each individual section can take hours or days to acquire. Multiplied by the large section numbers needed to cover a three-dimensional volume with good axial resolution, this means that a 1 × 1 × 1 mm block can take many days to weeks in acquisition time alone to produce a dataset. An additional challenge is the enormous amount of data generated by spatial omics technologies. Spatial omics methods, even when operating in 2 dimensions, routinely produce datasets containing tens to hundreds of thousands of cells, each possibly described by tens to hundreds of different measurements. Beyond the computational infrastructure necessary to process and store this volume of data, the amount of information produced by these methods can be overwhelming. This issue is heightened for three-dimensional data, since exploration of this data is invariably performed on two-dimensional surfaces. The ability to visualize high-dimensional, complex data is a key element to enabling full exploitation of any spatial molecular profiling strategy. Approaches to address such issues include the use of dimensionality reduction to visualize a dataset in a simplified format or exploitation of machine learning to recognize spatial patterns. However, in both cases, the ability of an individual to explore a dataset naively is greatly diminished. Several software packages exist to facilitate visualization of spatial omics datasets. Some have arisen as spatial add-ons to widely used single-cell analysis packages, such as Sara and Scanpy, and some developed from scratch with spatial analysis in mind, i.e. Giotto, Vitesce, STViewer. However, none of these can handle three-dimensional data natively. Virtual reality, VR, and its cousin, augmented reality, have significantly matured in the last few years and are set to revolutionize the gaming and entertainment industries. These technologies are increasingly applied in industrial design, medicine, healthcare, and education. However, application of VR to the exploration of scientific data has lagged behind. Indeed, VR has just started to be applied to biological data, mostly to explore gene networks, to annotate neuron morphology, or to visualize microscopy data, both academically, i.e. VLoom, CellExcel VR, Terra VR, Confocal VR, and commercially, Aravis in VR. Here, we describe a VR visualization and analysis platform for spatial multi-omics data. This virtual laboratory, Theia, so named after the Titaness of Sight in Greek mythology, provides a multi-user, three-dimensional environment for exploration of spatial datasets. This platform can accept segmented data with large numbers of molecular or other annotations, project them in 3D, and enable their intuitive manipulation and visualization of patterns of gene or protein expression. Theia is also enabled for on-the-fly analysis, for example with integrated functions for dimensionality reduction that can be used to identify groups of cells for reprojection into the 3D object. Additional analysis capability can be easily added to customize the platform through a convenient application programming interface, API. Theia operates using the Unity VR engine in the Steam VR environment and is therefore compatible with a variety of consumer VR devices and accompanying high-spec personal computers. To illustrate the utility of Theia for visualization and exploration of 3D datasets, we generated 3 segmented, molecularly annotated samples, Table 1. 2 biopsies from human breast cancers, in the sub-m size range, were analyzed by serial sectioning followed by IMC and realignment of the processed imaging data. The third dataset is a large-scale model representing a 300 micrometers thick section of an approximately 1 centimeter tumor from a syngenetically transplanted mouse triple-negative breast cancer, TNBC, model. The latter model was produced by serial block phase 2 photon imaging, serial 2-photon tomography, STPT, followed by IMC on recovered sections and contains over 2 million cells. While Theia was designed for segmented molecularly annotated datasets, it is also compatible with a wide variety of different data sources, ranging from segmented, 3D lightsheet microscope data, including time course data, to non-spatially resolved single-cell RNA-seq datasets, which can still benefit from the ability to visualize the dimensionality reduction plots in 3D. Any single-cell level data can in principle be visualized in virtual reality, benefiting from the added sense of depth, a simplified user interface, and a large visualization space. Results. The IMAXT pipeline for spatial analysis of biological samples. The imaging and molecular annotation of xenografts and tumors, IMAXT, CRUK Cancer Grand Challenge project is a collaboration between research groups located in the UK, USA, Canada, Switzerland and Ireland. The goal of this consortium is to enable the production of 3D multiparametric models of breast tumors in order to increase the current understanding of tumor progression, dissemination, and resistance to therapy, and to pave the way for the development of better diagnostic tools and treatments. At the core of the project is a sample processing and analysis pipeline, Gonzalez-Solares, EA et al. 2021, Nature Cancer, submitted, that our consortium developed, combining single-cell genomic, transcriptomic methods with large-volume 3D imaging via STPT and in situ molecular profiling, Figure 1. Samples that enter the pipeline within IMAXT can come from mouse models of breast cancer, obtained from genetically engineered mouse models, GEMMs, from tumor cell lines injected into syngeneic hosts, or chemically or virally induced tumors, patient-derived xenografts grown in immune-compromised mice, or primary human biopsies or resections. As a first step, one or more fragments of each sample are collected and cryopreserved, and used, after dissociation, for single-cell RNA sequencing, usually using the 10x Genomics platform, or single-cell whole genome DNA sequencing via DLP+. The results of these are used to produce a survey of the cell types and cell states present in the sample, and to guide the development of the gene and antibody panels used for in situ profiling. Since the data yield of disaggregated methods is presently superior to that of in situ technologies, this step is very important to ensure the spatial analysis can capture the most relevant parameters, unless a good set of curated markers is already available. The bulk of the sample is then embedded in an agarose block and subjected to STPT imaging. This method produces a distortion-free 3D image of the sample with up to 4 fluorescence channels. This instrument images a plane immediately below the block surface and then cuts a thin, greater than equals 15 micrometers, section from the sample to enable imaging of the next plane of the sample. In short, STPT combines a vibratome and a 2-photon microscope. STPT presents 3 key advantages over other whole organ images modalities such as light sheet microscopy. 1. Image quality and resolution don't change with imaging depth, since imaging always happens within a short distance from the top surface of the block. 2. Objects of any size and depth can be imaged, with an adjustable axial resolution, physical slice thickness. 3. The sample is sectioned, greater than equals 15 micrometers currently, and imaged at the same time, producing sections that can be further annotated. These gather in a collection vat in the STPT instrument and are recovered, in random order, on glass slides or in a known order with an automated collection device, currently limited to sections of greater than 25 micrometers. In our workflow, sections are counter-stained with nuclear and membrane dyes and re-imaged using a whole slide scanner. Each slide image is then registered to the 3D volume produced by 2-photon imaging. To facilitate this step, we co-embed spherical agarose beads coated with green fluorescent protein in the sample block. The cross-section of the beads produces a unique pattern for each slice, which can be easily segmented and matched across imaging modalities. Gonzalez-Solares, EA et al. 2021, Nature Cancer, submitted. Collected sections are then available for further molecular annotation using spatial transcriptomic or proteomic methods. Generation of an integrated dataset from a mouse model of TNBC using STPT and IMC. The 4T1 cell line is a widely used model for basal-like, Claudin-low breast cancer. It can be syngeneically transplanted in immune-competent BALB, C animals or introduced into immune-compromised recipient mice. Prior work has indicated that 4T1 is both genomically and phenotypically heterogeneous, and we previously identified subclonal populations of 4T1, clones E and T, which can selectively form vessel-like networks when orthotopically transplanted. We therefore used this cell line to create a demonstration model for THEA. We fluorescently labeled the parental 4T1 cell line by expression of TD tomato and labeled the 4T1T subclone by expression of GFP. We orthotopically injected a mixture containing 80% parental and 20% clone T into an NSG recipient animal and resected the tumor after 21 days, when its diameter had reached approximately 1 centimeter. We imaged the entire tumor using the STPT and collected 20 consecutive sections for further annotation, corresponding to a depth of approximately 300 micrometers. These sections were imaged on a slide scanner, stained with a panel of 33 metal conjugated antibodies and imaged by IMC. After co-registration and segmentation, we obtained a 3D model with an axial resolution of 15 micrometers, roughly an individual cell diameter, and over 2 million unique cells mapped over a volume of approximately 12 millimeters. Even in the 3D STPT image alone, we noted different spatial distribution for clone T as compared to the parental 4T1 cells. Figure 2a, supplementary video 1. The former distributed in foci within the tumor mass, while the latter were largely uniform. Being able to combine this imaging with IMC revealed that clone T gathered often in proximity to necrotic areas with substantial myeloid infiltration. Figure 2bd, S1, supplementary video 2. Interestingly, tumor cells expressing the proliferation marker Ki67 often had low expression of the TD tomato fluorophore. Figure 2f. We could also identify host vascular cells expressing endothelial markers, CD31, CD34, and parasite markers, PDGFRB, Desmin, alpha SMA, stromal cells, and cell clusters characterized by high expression of the M2 macrophage marker CD206, MMR, figure 2c, E, figure S1. In addition to cell types, we were also able to interrogate cell states. We could clearly identify hypoxic cells, both in the TD tomato plus tumor compartment and in the GFP plus 1, as well as cells characterized by high amounts of phosphorylated S6, SR235-236, ribosomal protein, a downstream target of the mTOR pathway, figure 2f. Overall, our results indicated a clear spatial organization of the tumor, with distinct populations of tumor cells occupying discrete neighborhoods. In general, GFP plus neighborhoods were characterized by low proliferation, low expression of epithelial markers, and high levels of hypoxia-related proteins, i.e. GLUT1, carbonic anhydrase X. These cells also had higher expression of CD44 than the parental 4T1 cells, figure 2f. All of these characteristics can be interrogated in 3D in virtual reality using Thea, figure 2g, see below. High-resolution 3D datasets from human cancer by serial sectioning and IMC. As additional input to our virtual reality platform, we have also provided 2 human breast cancer samples that have been previously generated as part of IMA-XT consortium efforts. These models were generated from a HER2+ invasive ductal breast carcinoma biopsy using 3D IMC to achieve high axial resolution together with in situ molecular annotation. Kuett and Catena et al. Nature Cancer, submitted. Briefly, the 3D IMC method is based on serially sectioning a paraffin-embedded tissue sample into 2 micrometers sections prior to sample staining and acquisition. This is followed by computational image processing to achieve the final 3D model with a voxel size of 1 × 1 × 2 micrometers, XYZ. The first model has the final dimensions of 652 by 488 by 304 micrometers, and the second one 606 by 686 by 184 micrometers and comprise 108,000 and 81,000 segmented cells, respectively, figure 3a, left and right respectively, providing an interesting substrate for exploration in VR, figure 3bf. Notably, these models reveal spatial distributions of marker expression that are not evident in 2D images, thus illustrating the ability of 3D models to raise interesting hypotheses. For example, these models show a varying pattern of CK5 high and SMA high epithelial basal cells, a dense colocalization of CD8+ and CD4+ T cells near endothelial cells, and lining of PS6, SR235-236, high cells along the epithelial tumor compartments, figure 3CE, S3, supplementary video 3. In the larger 3D IMC model, clusters of potentially invasive cells at the tumor periphery have been captured, and the 3D visualization makes it apparent that these clusters are disconnected from the main tumor bulk. Figure 3F. Development of a virtual reality laboratory to explore spatial molecular data. To enable exploration of three-dimensional, molecularly annotated datasets, we developed a virtual reality implementation that takes lessons from the computer gaming industry and applies these with the goal of enabling intuitive spatial analyses of cellular arrangements and gene expression patterns in biological samples. VR has the advantage of not being bound by the dimensions of even a very large computer monitor. Instead, users can literally step inside the data, generally in a roughly 5 meters area, and apply different analysis paradigms and explore the outcomes on the fly. Finally, our implementation creates a multi-user environment, such that investigators can convene in a virtual space to interrogate collaboratively high-dimensional datasets. We named this implementation Thea. Thea has the potential to impact the analysis of spatial data analogously to the impact of the development of genome browsers have had on our ability to interrogate and understand next-generation sequencing datasets. Thea was developed on consumer-grade VR headsets, any compatible with the SteamVR system, and computer hardware to make it accessible to any laboratory. The investment needed for operating the software, including headset, controllers and computer, is less than the cost of generating one single-cell dataset. The software is built upon a popular and well-supported game engine, Unity, which results in broad compatibility. Our primary concern was to create a space that was comfortable to work in for several hours with tools that responded quickly and usefully. Additionally, it was imperative for us that no programming be required and data analysis be achieved with simple visual metaphors, to make the learning curve as shallow as possible for new users. The objective was a balanced design, both scientifically functional and with limited visual clutter. Figure 4A, supplementary videos 4-5. Thea tools have a universal language which is easy to learn. They consist of two varieties: surfaces for generic analysis, displaying and highlighting cell populations, or visualizing gene expression, and cabinets for specific analysis such as dimensional reduction or differential expression. Grabbing and placing of objects is the main mode of interaction. For instance, the sample can be rotated and zoomed just by grabbing it and moving it closer or farther away. Supplementary videos 4-5. Additionally, pointing, buttons, sliders, and levers are utilized to change visualization parameters such as cell size and scale. Tools for moving, selecting specific cells, pointing and taking screenshots are housed on the user's wrists. Figure S2A. This ensures easy access from anywhere within the simulation. Features of the dataset to be analyzed, populations, genes, cell types, are displayed as VR tiles, which the user can interact with by moving them in specific slots into the environment tools. See examples in figures 4, 5, supplementary videos 6-7. These interactions are subtly guided by objects snapping together when in proximity of a compatible slot, and by haptic feedback through the VR controllers. Subtle animations and in-world screens are also utilized within Thea. This prompts the user toward correct actions and ensures simulations become intuitive. Figure S2B, supplementary video 8. Interactive data analysis in the VR environment. Thea is designed to analyze samples formatted as cell catalogs, with each cell characterized by a series of spatial coordinates, corresponding to either real space or embeddings along dimensional reduction axes, and parameters, gene, protein expression or morphological measurements. Cells can be pre-assigned to different populations, for instance by means of clustering methods such as the Louvain or Leiden algorithms, or populations can be defined directly by the user by painting over cells with a specific tool. Thea uses a dedicated data storage format optimized for small size and speed of loading, but can import data from most single-cell processing data formats, e.g., the one specified by the AND-Data Python package used by Scanpy, or R's single-cell experiment in Sara. Information about the sample is displayed through dedicated panels, which display details at the sample level, general metadata on sample generation, size, features, etc., Figure 4D, or at the population level, or single-cell level. For the latter two, the panel shows a customizable maker expression heatmap, which allows users to get a general view of gene or protein expression in a population or in a given cell. The information displayed in the panels changes dynamically as the user selects different cells. Figure 4D, Supplementary Video 13. Advanced Analysis and Plugin Structure. Compatibility of Thea with other data types. We expect spatial datasets, such as the ones presented here, to become increasingly available. However, true, molecularly annotated, 3D datasets are still relatively rare and difficult to generate, requiring substantial infrastructure and effort in technology implementation and data analysis. We therefore have also made Thea compatible with the analysis of datasets and data types that are already widespread. Disaggregated single-cell analyses, transcriptome, copy number, methylome, etc., are increasingly used in all areas of biology, including to interrogate the tumor microenvironment and the clonal evolution of different tumor types. The technology to generate many of these data types is now mature and broadly available. Thea is compatible with all single-cell datasets, and includes a data converter for SCANPs and data objects, one of the most popular formats. Other formats can be loaded if converted first to AND data, which is usually a straightforward process. If positional information is not included, the dimensional reduction embeddings, TSNE, PCA or UMAP, are used in lieu of the spatial coordinates for visualization, and populations, clusters or mapped to cell types. All of Thea's tools, on-the-fly dimensional reduction, differential expression, gene averages, etc., are compatible with single-cell data. As an example of the usage of this data type, we provide with Thea a single-cell dataset obtained from the same tumor type and stage represented in our STPT, IMC 3D model. Figure 6, Supplementary Video 22. Whole mount microscopy is another family of technologies that has recently seen increased democratization, in part thanks to the development of novel microscopy techniques such as light sheet, Bessel beam, lattice light sheet, STPT, and others. Thanks to technical advancements, whole organisms can now be imaged in toto, including live imaging of their development. Datasets of this type have been published for D. melanogaster and zebrafish early development and for pre-implantation mouse embryos during gastrulation and early organogenesis. In parallel to the technical improvements in imaging, improved methods for object segmentation have allowed the identification of every cell in these models, and their tracking across time. Since the output of these processes is a catalog XYZ position for each cell at different time points, Thea can be used to visualize them. To enable visualization of time series data, we modified one of the functions of our viewer, normally used to cycle through the different cell types present in a sample to be compatible with time points. As a result, Thea can be used to load and visualize spatial time-lapse data. Cell labels and markers, if present, can still be visualized as for other samples. As an example of this, we included with Thea a pre-packaged version of the mouse development dataset from McDowell et al. Figure 7a, Supplementary Video 23, for exploration. Discussion. Methods. Sample generation for the 3D STPT sample. Fiducial beads conjugation for STPT embedding. STPT sample preparation and STPT imaging. Slide imaging. Images were captured post-STPT with the Zeiss Axioscan Z1 microscope slide scanner with a resolution of 0.44 micrometers pixel. GFP. Excitation filter 465 to 490 nanometers. Emission filter 460 to 480 nanometers. Exposure time 250 milliseconds. TD Tomato. Excitation filter 545 to 565 nanometers. Emission filter 578 to 640 nanometers. Exposure time 700 milliseconds. The surface area of each section was imaged including the GFP plus fiducial marker beads surrounding the tissue. IMC antibody conjugation and panel preparation. IMC sample preparation for the STPT, IMC dataset. IMC image acquisition. STPT image stitching and Z-alignment. Because STPT is the modality that has access to a relatively intact sample cube, STPT images serve as the anchor for data processing. It was important then to ensure that these images provide the most accurate representation of the sample possible. In the case of slice-to-slice registration, a simple nearest neighbor search was enough to find the bead pairs in contiguous slices, and by identifying in which slices a given bead appeared, due to their size, usually 3 to 5 slices, We could use the measurements of their central coordinates to align all the slices in the sample. Typical alignment errors were of 3 micrometers, while the action of the microdome introduces a drift that from top to bottom would skew the misaligned cube close to 20 micrometers in one direction. STPT to whole slide scanning to IMC registration. Typical registration errors, as measured by comparing the bead centers, are of 6 micrometers for the IMC to STPT registration and 7 micrometers for the Axio to STPT1. IMC segmentation. Dataset assembly and single-cell analysis for the STPT, IMC model. 3D IMC human breast cancer models. Single-cell dataset processing. Approximately 1,800 cells were identified as high quality after removing doublets, cells with low counts, cells with high mitochondrial reads and cells with high ribosomal reads. Cell types were identified using the CellAssign R package and a custom annotated marker matrix. Each cluster was further validated by performing unbiased clustering with the Leiden method and verifying that each CellAssign cluster corresponded to one or more Leiden clusters. Normalization, log transformation, scaling and dimensionality reduction, neighborhood analysis were all performed using the ScanPy package. Mammary duct volumetric dataset. The mammary gland sample was obtained from a virgin NSG-GFP mouse, Jackson Labs, NOD. CGPRKDCSCID-IL2RGTM1WJLTG, CAGEGFP, 10SB, SZJ, stock 021937, approximately 2 to 3 months old. The animal was first euthanized using increasing concentration of CO2 as prescribed by the Schedule 1 of the UK Animal Scientific Procedures Act, ASPA, and specific in the Home Office approved project license mentioned above. The inguinal fat pad was dissected and